Welcome to my YouTube channel. Today we are going to talk about DNA methylation analysis and the different methods. DNA methylation analysis can be done using a conventional PCR technique where you use two sets of primers, one for methylated and the other for unmethylated DNA sequence, and the result can be viewed on gel electrophoresis. The other methods are high resolution melt curve analysis and methylated DNA immunoprecipitation. Why DNA methylation is very important is because of the fact that 80% of the cytosins in CPG regions has been linked to human cancer. So we are interested in knowing the methylation status of few cancers like mainly CNS neoplasms. So for this, the basic principle is bisulfite sequencing. This is commonly used to identify individual methylation sites and quantify the level of methylation in a specific region. This protocol converts unmethylated cytosine residues to uracil, while methylated cytosines remain unmodified and are analyzed through downstream PCR or qualitative PCR or sequencing. High resolution melt curve analysis is the qualitative analysis of a DNA fragments melt curve following PCR amplification. It's a simple, cost effective, and rapid tool for studying genetic variation with applications in single nucleotide polymorphisms, mutation scanning, species identification, and also methylation analysis. So here today we are discussing methylation analysis. In this method, as I said in the previous slide, DNA is first treated with sodium bisulfite to convert unmethylated cytosine residues to uracil. Then we do PCR cycle. We run a PCR cycle for amplification of the converted DNA, unmethylated DNA as well as methylated DNA. And methylated DNA and unmethylated DNA both have unique melting temperature. So due to difference in the melting temperature of the unmethylated and methylated DNA, this technique is used for the identification of methylated and unmethylated DNA in your DNA sample. Methylated DNA immunoprecipitation technique, it is used to examine genome-wide changes in the DNA methylation patterns across various cell types. In this technique, DNA is isolated from cells and DNA extraction is done by uh, physical extraction method, that is sonication. Antibodies specifically targeting methylated DNA fragments are used to isolate methylated regions. The methylated DNA fragments can be identified using high resolution DNA microarray or NGS. So basically, as the name suggests, we are using antibodies against methylated DNA fragments. Thank you.